Welcome to the Microscopists, a bite-sized bio podcast, hosted by Peter O'Toole, sponsored by Zeiss Microscopy. Today on the Microscopists.
Today on the Microscopists, I'm joined by Graham Wright from A-Star, and he talks about changing from being a PhD pure research into running a core facility and what motivated him to do that.
Over time, we sort of started developing it more and more in the kind of core facility model. So we started to share those instruments with the wider audience, neighbouring institutes or the wider A-Star.
Also, what it's like switching from a career in the UK to Singapore and all the advantages that's enabled him to achieve.
It's got historic links to the UK, and English is the mainly spoken language. There's 4 official languages, but you find with English everywhere in Singapore. So it's quite an easy place to come and either to visit or to certainly to live and establish a kind of life here.
Plus, we talk about his hobbies and how he balances his family, both in the UK and over in Singapore as well.
I think the realities of going in winter, if we've been a few times for Christmas, obviously it's a bit of a shock. But even that, the novelty of being cold and when you breathe out and the steam comes out, condensation comes out, they're fascinated by those sort of things because they never get exposed to that here.
All in this episode of The Microscopists. Hi, welcome to The Microscopists. Today I'm joined by Graham Wright from A-Star over in Singapore. Morning, Graham. Or evening, Graham.
Good afternoon, good morning to you. Glad to be here.
So this is like quite a lot of people I've spoke to who have gone sort of which direction, east to west. But you're one of the few scientists who've gone west to some way east itself. What inspired you to go to Singapore?
It's a good question, and as you know, and maybe we'll get to, I've been here for quite a long time, but it was a chance opportunity to a large degree. I was studying for my PhD in Edinburgh and looking to what to do next. And my supervisor at the time, Nick Reed in Edinburgh, connected me to someone who was interested in looking for a microscopist of some sort, or someone to run a microscopy facility in Singapore. And kind of the rest is history. It was a chance encounter, a meeting at a conference with this person, Greg Jed, and that led to an opportunity to come visit Singapore and later to run a facility at Temasek Life Science Laboratory where I first started out here.
So what year was that?
That was at the end of 2007 that I first moved here. So 16 years ago, which is frightening to think about that I've been here for so long.
Do you have children at that point?
Nope. The plan during my PhD was that I would move down to London, where my then girlfriend was living, if opportunity allowed. And fortunately, I persuaded her to come join me in Singapore, take a chance. Now married to that same girlfriend, Charlotte, and we have 3 children together. But no, it was a much easier logistical move moving here back then.
So we'll come on to that then. So what was your degree in?
My bachelor's was in plant sciences, which was also at Edinburgh, but it was the way the biological sciences are structured at Edinburgh. You go in as biological sciences, and then you can specialise into the 3rd and 4th year of the degree course. So I went in not knowing which of those honours years to do, and fell in love with plant science, but specifically mycology within that, which was the study of fungal cell biology or fungi as a whole. Partly because of microscopy, because the lecturer, which was Nick Reed, who I made reference to earlier, was using confocal microscopy and imaging these incredible kind of dynamic processes in filamentous fungi in their hyphal networks. And was showing this data when he was teaching on the botany course or on the plant sciences course, and showing this microscopy data in 2D and in 3D and in live cell imaging. And that really sparked a passion for that subject, for mycology, but really for the application of microscopy to that subject, which is what led kind of fortuitously to my PhD project in that group.
So you stayed in your undergraduate to your PhD at the same place then?
Same place, yeah. But obviously changed a lot in that. So my undergrad was plant science as a whole, but that kind of ignited my interest, which through conversations with Nick, through my final year project and so on, sort of led to the development of a PhD project that he'd kind of put together as a collaboration between the physics department in Edinburgh, who were optical physicists implementing and inventing novel microscopy techniques and trying to find biological applications for that, which was all centered around his fungal cell biology interests. And that's really what defined my project, was application development for laser tweezers or optical tweezers, and how we can use them to study fungal cells in different ways, everything from how they grow to how they germinate, to how they communicate with one another. So it was really the tools were developed, of course, but it was really finding novel applications for those tools.
So technologically speaking, that's super geeky.
Yeah, a little bit. I mean, there were some incredible optical physicists at Edinburgh who were putting these systems together. So I can't ever claim to be an engineer or a physicist, but it was that, they were building systems that were not generally that accessible to every biologist. But I think I was being encouraged to be a microscopist from that early stage and get in there, work with them, understand the systems, and make use of them. So I had a supervisor in the physics department and a supervisor in the biology department, and I would have to have supervisor meetings with each of them. And it was great if they were on their own, because I could talk about biology to the physicist, and I could talk about physics to the biologist. When they were in the room at the same time, it's a bit more dangerous because they could both sort of probably understand it more than I could in both domains.
So I was going to ask about how you found having two split supervisors. How often did your physics supervisor drive you in one direction, but the biology one then pull you back and take you back in a different direction? How was that fairly harmonious, or was there sometimes you cherry pick maybe what you liked one was saying and what the other was saying?
Yeah, it's a good one. I was kind of in the middle. It was a pretty harmonious relationship. There was interest on both sides because of the sort of novelty that the biology brought to the physics department and vice versa. I don't remember many particularly difficult moments. I think it was an enthusiasm for the project because of perhaps the novel outcomes that were going on through it. And as I said, some great physicists. There was one standout, Jochen Alt, who was really building these systems, and I would interact mostly with him. But I had to give institutional seminars, internal seminars at both forums, which was always threw up all sorts of different questions, as you can imagine, from two different disciplines.
So I have a question then. In your current job, do your users think you are a biologist or a physicist? Do they ask you, "Oh, did you do a physics degree or did you do a biology degree?"
I think I've never asked, but I assume they think I'm a biologist, and the conversations we have are around biology, not so much physics. We're not in the facility that I run out here in Singapore is the A-Star Microscopy platform, which is a central core facility, as you know. We're not building and developing novel microscopy tools or building sort of a kind of pre-commercial release system. We're exclusively housing commercial microscopes. So it's perhaps I come across less as a kind of physicsy person because of that.
I think it's fine until you start training them sort of the depths of confocal or the depths of super rares or the depths of FRAP, and at that point, that's when they go, "Oh, did you do a physics degree then?"
Probably strive to keep it digestible, both for my own sake and for theirs. That it's it's often the conversations we have with our users who are predominantly biologists are about how to use this instrument for my experiment, how to get the most out of it, but without going into every nuance of the kind of technical details around that.
Yeah, I don't think I go into the fine nuances. I just think that they think I'm such a bad biologist. I must have been a physicist, and the physicist is a bad physicist must have been a biologist. So I think I'm hiding somewhere in the middle.
I think you'd have to ask the users probably for the inner truth on that one, actually.
So, I'm going to take you way back. So when you were, do you remember the first job that you ever wanted to do as a child?
Yeah, I think growing up, my parents would probably back this up. I think I always said I wanted to be a pilot.
Mm-hmm.
And in my mind, from memory, it was a civil aviation pilot, and I had this kind of very kind of romantic dream of what it was like. I for whatever reason, that I pursued my interests usually when it came to school and studying and so on. So I pursued my interest. I loved biology always at secondary school. I was actually torn between biology and geography when it came to A levels and applying to university and so on, and chose biology. But yeah, so I pursued my interest in what I was enjoying at the time, what I was sort of thriving off as a topic, and I don't think biology naturally leads to being a pilot. So I went off on this tangent of plant sciences, then mycology and microscopy.
So where did the plant science come from?
Yeah, good one. So as I said, Edinburgh, structure of the degree at the time, at least in Edinburgh, was that you entered biological sciences, and then through the choices of courses that you did, that sort of limited the honours years available to you. I think there were 14 honours years when you first went. Oh, it's a Scottish university, so it's an extra year. So you had 14 available to you, and then as you narrowed it down, you got less and less. But I was interested in the mycology, as I explained earlier, and enjoyed the plant sciences a lot. Not so much from the ecology side, more, or botany side, more from the kind of the sort of cell biology level of plant science. And there were some excellent teachers and faculty and research going on in Edinburgh in that space at the time, which was really kind of motivating and inspiring. So I again followed my interests, which is perhaps the story of my career in many respects.
Okay, so you did your degree, then on to the PhD, as you said. Did you go on to a postdoctoral?
I didn't. So during my PhD, I kind of made the decision that I wanted to go into microscopy and facilities management, and specifically. And at the time, that was kind of quite a newish thing to do. But my lab had spawned a few people. So Sam Swift had gone to Dundee at the time to establish a facility there with Jason. And a few others had gone and either worked closely with or been associated with microscopy facilities. And I was very passionate about the microscopy. I'd got involved in teaching people how to use the confocal that we had in the lab from other labs in the department, and sort of set my sights on this being a potential opportunity. And then, as I explained, the right chance came up at the right time. It just happened to be halfway around the world in Singapore. So yeah, I was quite focused. I made a clear decision not to pursue a postdoc. I think I didn't like how narrow I was getting into one particular topic with a PhD, but I did enjoy working with lots of other different people and using the common tool set, as it was, the confocal, for studying all these different organisms, different problems and challenges, largely around plant biology, because that's the department I was in. But yeah, thrived off that. So a few people had sort of trodden the path ahead of me, and then opened up my eyes to this opportunity, and then was lucky, I guess, with the timing.
Yeah, I think 2007, it's quite foresightful to think that's what you want to do and then find a job in it. Because you say it was the core facilities back in 2007 were only really emerging. They were only just starting to gather a bit of momentum. So to know those jobs that existed at a PhD level is quite impressive. And you say, so yeah, Nick's lab and
I think Nick's lab was a lot of it. He'd got me involved with the Royal Microscopical Society, of course, and I think that's where I first met you, was a microscience, as it was called at the time, down in London in Excel. And that again, that opened my eyes to, there was a microscopy community out there. There was a community of people that was teaching other people how to use microscopes or facilitating access to advanced microscopy. So I'd sort of my eyes had been open to it in that sense. Lucky that the opportunity came up at the right time. I was then very, very lucky. I secured the job in Singapore. And there was I had to wait a period of time while they worked out the visas and the employment pass and so on. And at the time, the director of the institute I was moving to was also changing over. And Steve Cohen from Emble was moving to be the director of this institute in Singapore. So he helped arrange for me to visit the ALMF in Emble for a couple of months en route to Singapore, if you like. And I spent a great time there. Stefan Taeyong was there. Timo Zimmermann was just leaving, I think, at that time, or was on a visit back from Barcelona when he was going there. And it was a fantastic opportunity, right? I got to see how, one of the early facilities and one of the most developed facilities at the time was running, and how well supported it was. Kota was in the neighboring room doing the image analysis and that side of things. So really, that helped set me on a path as I arrived in Singapore to try and emulate that in some way, or at least bring the relevant bits of that to the facility in Singapore.
3 mega stars of core facilities there, aren't they, Stefan?
Yeah, well, exactly. Yeah, Arne Seitz was also there. I think he was on his way to EPFL at that time. So lots of people there, but also that network, right? The people I've just listed have been phenomenally helpful to me. When I arrived in Singapore, I could ask them, for advice and knowledge. Rainer Peppercock, of course, was there overseeing ALMF at the time. So some brilliant connections, and subsequently had lots of chances to interact with that community at other meetings or in opportunities to travel and so on.
So thinking about, we'll come back to that in a moment, actually, because I know you see more Rainer and Timo in different meetings now. But when you moved to Singapore, what did you think you'd miss most?
From a personal side or a cultural side. What did I think I'd miss most before I left? Yeah. I was really worried you couldn't get Yorkshire tea here for a while, but it turns out you can, so that's okay. And I remember, but as I said, my then girlfriend, we were sort of exploring whether you can get this, that, and the other. So it was comfort foods and that sort of thing. The reality is it's very easy to live in Singapore. It's an incredible place, and you can get most things from most different cultures and parts of the world, but also the food that is produced locally or served locally is phenomenal. So I think in truth, probably in hindsight, the miss the most is probably family, right? Because it's a long way. Yeah. There's a big time difference, 8 hours, as you know, at the minute. We're sort of a long way from home, but things like it was Skype originally, now Zoom and FaceTime and so on, it makes it much easier to be this far away from home, because you get to see one another and have these kind of conversations, which is quite different to an isolated phone call.
And how often do you get back, or how often do your parents come over visit you?
So pretty much I'll go home once a year. Maybe if I'm lucky with a conference that I'm attending in Europe, I get another chance. So once a year, generally in summer. Winter's a bit of a shock coming from a tropical country when you've been away for so long. But yeah, once a year I'd go there, and my parents, on average, I guess, have visited here once a year as well. So still plenty of visits, but it's not quite down the road, right?
Yeah, and quality visits, because they're longer, probably periods of time.
Yeah. So it's in that sense, it's a long way away, but we sort of maintain that.
And your parents are Manchester way?
Yep. Yeah, I grew up in South Manchester, and they're still there in Sale. So that's still home. My wife's parents are based down in Farnham in Surrey, which is obviously a bit of a distance away, but also accessible.
Yeah, but if you go and visit your parents, and from Singapore to Manchester, not only cold but continuously damp, not just heavy downpours.
Yeah, no, it's not so short and sharp in Manchester. It's sort of long and prolonged.
And for Mancunian to actually say you want Yorkshire tea, is that allowed?
Definitely allowed. It's definitely the best tea, for sure. Don't think there's
between Yorkshire.
I think there's an equivalent of it on the other side.
And so, when you went to Singapore, what did you find most challenging on a personal level rather than work?
Honestly, I think it's the heat. I'm physiologically not designed to live in a tropical country. I'm born and bred in Manchester, as we've discussed, and I think I'm much more suited to that climate. In terms of cultural things to adjust to, as I said earlier, it's really quite an easy place to assimilate into. There's a mix of everything here. It's a very kind of diverse place. Obviously, it's got historic links to the UK, and English is the mainly spoken language. There's 4 official languages, but you're fine with English everywhere in Singapore. So it's quite an easy place to come and either to visit or to certainly to live and establish a kind of life here. So there wasn't that much that was challenging, apart from it was just really hot and sweaty the whole time. It's continually, over 30 degrees and 100 or so % humidity. So it's quite a strong climate. Obviously, it's air conditioned inside, so that helps.
Yeah, inside. But one of your hobbies is triathlons. You sent me a picture, which is you with your, I presume, triathlon.
They're my kids. Yeah,
certainly a road bike and your children. How do you train then if you're not don't like the heat and you're in Singapore? It's quite warm.
Yeah, I think the when you're exercising and you get hot and bothered, that's okay because you're exercising. It's more that I find the heat a problem when you've got to try and rush to a meeting and look reasonably presentable, and that's difficult to do. So I don't mind getting hot on a bike or running or whatever, but it's you don't want to plan to arrange an important meeting an hour later because you're going to still be very red-faced and attempting to cool down for a long time.
How do you find the endurance aspect though? Because so certainly when I came over Singapore, running was great fun for 10k.
Yeah,
realize actually going more than 10k is I was going to start to feel it. So how do you
it's hot, and so the secret. So I cycle mainly here. I do some triathlons, but mostly I cycle, and I've got a great group of friends here that I cycle with the weekends. And the secret is to go early, so you get home before the heat of the day kicks in and before the traffic wakes up and the roads become a bit more kind of chaotic. But we go early. We meet at 7 o'clock in the morning. We're home by 9 o'clock in the morning, and with that you can almost do a decent loop of Singapore because it's not a very big place.
Yeah.
That's how we deal with it. So I wouldn't ever go for a bike ride if I could avoid it in the heat of the day. It's a much less pleasant experience. And then carry as many water bottles as you can conceivably squeeze on the bike. Is the other secret, which for running is very hard, right? Because you tend not to run with a lot of water, and it's extremely dehydrating.
So we saw your children in your previous picture, and here's a picture of your family.
Yeah.
For your children, how old are your children now?
So Hartley in the middle there, he's 11 years old. Then Connie near me is 9, and Otto has just turned 6.
I suppose they've only known life in Singapore.
Absolutely.
What do they make of it when they come back to the UK?
They love it. I mean, it's always a visit to the grandparents' houses or to see friends that maybe moved from Singapore or otherwise friends and family. And as I say before, we always go in summer, so the weather's usually pretty clement. And they have a great time, and it's all a big adventure. I think the realities of going in winter, if we've been a few times for Christmas, obviously it's a bit of a shock. But even that, the novelty of being cold and when you breathe out and the steam comes out, the condensation comes out, they're fascinated by those sort of things because they never get exposed to that here. And a funny one is that here, if your windows condense up, it's on the opposite side to what it is at home. It's on the outside rather than the inside, because the heat is obviously reversed. But the things that they notice, things like that, is always fascinating to watch because they're just not used to a colder climate. And the vegetation's different, the trees are different, the kind of animals you see are obviously quite different. We don't really see many in Singapore apart from pets.
So you've been there now for 15, 16, 17 years.
16 at the minute, but on the way to 17.
How long did you think you would be over there when we left you?
I think we thought we'd be here for like 2 or 3 years and go and enjoy it and then probably come back. And we haven't, right? I've changed jobs once, changed employers once. I moved from Temasek Life Science Laboratory after 3 years to A*STAR, which is the Agency for Science, Technology and Research, where I still am, although my role has changed significantly over that time. And the jobs are typically based on a contracted period, so 3 years plus. And every time we've got to the point of renewing, it's been a no-brainer. We've chosen and wanted to renew and stay for longer. And then suddenly you look back and like, what? We've been here for 15 plus years. Yeah, it was never in the plan, but it's been a lot of fun, and I think testament to how much we've enjoyed our time here as well.
So you mentioned lots of role changes within A-Star. What have those changes been?
So when I came across to A-Star, I joined an institute called the Institute of Medical Biology, which is within A-Star, one of the biomedical research institutes, and I joined as their microscopy facility manager, I think was my title. And they had a microscopy facility called the IMB Microscopy Unit, which was overseen by a PI called Sohail Ahmed, who was a great mentor and person to work with, who helped had established this facility in a relatively young institute. And it was a bit of a core facility, but some of the instrumentation was very focused on his lab's own interests, which were around F techniques, FCS, FLIM, etc., and pushing those techniques for those applications. But it also had confocals that were shared with the rest of the institute, live cell imaging systems, and so on. So that's where I started in A-Star, and over time we sort of started developing it more and more in the kind of core facility model. So we started to share those instruments with the wider audience, neighboring institutes, or the wider A-Star, and then more broadly with the Singapore community. There's two big universities here, the NUS and NTU, that started using some of the instrumentation, tapping onto our expertise, particularly when it was a unique capability, of course. And then we started to charge and have to cost recover, and some of the other factors of running a facility started to come to bear because it was growing in scale and size and user base. And it sort of evolved over time, and I stayed doing that for many years, sort of growing the role, expanding to cover other techniques. We added an electron microscopy capability. We accumulated more novel techniques, super resolution, and so on. Then we started kind of merging some of these capabilities with other institutes' capabilities to make sure it was a kind of more holistic core facility or central facility. So in that time frame, my title changed a little bit, as well as did my roles and responsibilities as we sort of evolved the facility. And then more recently, I've changed, and I now sadly don't do much microscopy in terms of very hands-on actually using the microscopes myself. But I've transitioned over to an organization called the Research Support Center, which, as the name suggests, we're here to support research, and that takes various different forms. Everything from overseeing and making sure that core facilities, or technology platforms, as we call them, are well run and coordinated and available to the community. We also try and alleviate a lot of the administrative burden off the scientists that are running the facilities. We do their billing, for example, and some of the marketing, promotion, inquiry handling, things, front office and back office, the way we refer to it. And that's sort of more managerial kind of oversight role. And then RSC, the Research Support Center, does lots of other things. We run a consumables business, so we buy in bulk, the best price possible, and sell it to the research community to drive efficiencies and make sure that we're getting a good price for these goods. We run real critical central support services like a glassware washing facility and a sterilization facility, biohazard waste handling, and so on. And that's really kind of bread and butter facility that keeps the research moving. All these other supportive functions, very kind of operational, hands-on, but really driven by helping the researchers crack on and do some great research while we take care of some of the other tasks. But microscopy is still part of that, I'm pleased to say.
So are you still leading the microscopy side?
I am. So it's about to be transferred actually to another institute, but I'll still remain involved and on the steering committee and helping to advise and consult Xiao Ma, who now runs that facility at an operational level, together with the colleagues there. So I'll be a little bit further from it going forward, but I still teach on the courses and take part in some of the initiatives we run. We've got a national level partnership between the major microscopy facilities in Singapore, which is called Singascope, and that brings together this community. And that I'll still be very actively involved in that sort of coordination as well.
So this is interesting now. So your role, it's what's been really good to hear is how your career has been developed and how A-Star have helped you develop your career as well.
Absolutely, yeah.
So it's not for everyone, but you've been able to progress, and I presume these are upward steps.
Yep, absolutely.
Yep. But now you're kind of leaving the hands-on, leaving the need for technical expertise to an extent.
Yeah.
Because you now have others coming in. So does that mean the conferences you go to are going to change? So instead of being as you mentioned earlier, Microscopists, so Microscopy, MMC as it is now, which is in your hometown of Manchester now.
Yes, I know that,
yeah. Your Elmies, your things like that. Are you going to have to leave those behind and start going to the ABRF or the CTLS, called Technologies for Life Sciences, or even in the UK, the Technical Specialist Network? Are you going to migrate to those now and prioritize those over others?
It's been happening. I guess again with hindsight, I have kind of embraced those opportunities. I haven't been to ABRF yet, but I have been to CTLS meetings in the past. I think the last one I went to was in Ghent, which was a great meeting. But I was running up until now. I've been running that in parallel with attending microscopy meetings and these more kind of facilities or core facility orientated meetings. And I've added on to that the global bioimaging meetings, which they call the exchange of experience, which has been a fantastic community to get involved in. Thanks to thank you very much. There you go.
So you sent one of the global bioimaging meetings, which was in Singapore itself, wasn't it? In this case,
it was in Singapore. Yeah. So the Singascope partnership that I referred to earlier, which was a kind of Singapore-wide coordination of all the different light microscopy capabilities, gave rise to me getting involved in global bioimaging. And it was by the director of IMB that I referred to earlier was Birgit Lane at the time, and she was instrumental in introducing me to John Erickson, who's in the center right of that photograph, who was heavily involved with Eurobioimaging, had helped develop this global bioimaging initiative together with Jan and Ellenberg and Antje and others that were involved in the early days of Eurobioimaging. And through Singascope, we got tuned into this. I went to a few of their meetings, and then we were asked whether we'd be interested to host the meeting in Singapore, which gave rise to the one on the screen. And they're very different conversations. It's all around bioimaging of some sort, whether it's LM, electron microscopy, or the more kind of clinical, preclinical methodologies. But it brings together this community who many of whom are running and hands-on with microscopy, but also there's the facilities angle, there's the policy and funding angle. There's a lot of different interest in that group. And they've proven to be really great meetings. Lots of good people there on the picture that have changed. Is a quite different conversation as well to we have at some of the others. A bit like the satellite meetings we see at ELMI, the core facilities satellite meetings. It's similar conversations amongst these groups as well.
And obviously we've got the LM facilities meetings, which are early January, so still fit in with trip home as well.
Well, and I certainly advocate those to colleagues and so on, and I know some of my colleagues have been to join them, and that community is brilliant, right? Is again sharing experience of running a facility. We will have the same problems or same challenges in different kind of ways, but I'm a great advocate of sharing that, and visiting each other's facilities is always an extremely useful thing to do to get insights in how it's ran, how they approach different, the same problems in different ways. These sort of things is extremely valuable.
I think what's interesting as well is the problems. So you're now, I guess, not just microscopy. You've got other core facilities, by presumably in genomics, in mass spectrometry, and other technologies out there. And even though like global bioimaging, it's imaging, it's microscopy, it's for the life sciences, but I'm presuming that the discussions you have actually are completely relatable It's just the name microscope can be changed for genomics. The name genomics can be changed for mass spec. You know, you hear sometimes electron microscopy say how their core is so different to a light microscopy core. Actually, I don't think they are. There's a different bias, a different balance of instruments and staff ratios and time and demands. But ultimately, the fundamentals, the basics are the same. So those meetings are still super useful for no matter what your technology bias is.
Yeah, you can. It's very translatable. I totally agree with that. I think we see it is very, very kind of what applies here can apply there. I think what we see in this oversight or coordination role that we have is that the techniques are obviously very different. The way samples are prepared and processed and so on can be very different. We also have two sort of major different modes of operation. Some that are training followed by self-use of the instruments, or your supported self-use as it may be. But you know, they'll teach it to a user. It can be taught in a matter of hours or a couple of days, different sessions and so on. A way that you can get on and use the instrument at least at a certain level of proficiency. And that's perhaps typical of microscopy and flow cytometry to some degree, I think. And then we have other facilities or platforms that are much more kind of service orientated or collaboratively orientated, that it's not a case of we'll teach you how to use it and on you go. It's very much it will either do it for you or perhaps with you better in many cases, and really spend the time together trying to understand, is it this we need to do, is it this we need to do, or helping alleviate the kind of huge technological hurdle it is to get running on those instruments effectively. So coordinating that, obviously it's then different challenges that come with it, but I think appreciating that and appreciating that, okay, the expectations of this will be quite different to another facility is important, and somewhere we're trying to develop at the minute. And this is built off ideas coming out of ABRF and other publications from those sort of groups, particularly Phil Hochberger in Northwestern. We've had a lot of conversations with him, and Ian Smith, who used to be at Monash University in Australia. Is how to fairly kind of assess these platforms in terms of their performance, or at least set kind of expectations. And it's what we've come to realize, and it's obviously quite clear, is that they're not all working to the same aims, right? They all work in different models. They may, you know, publish a lot of papers as co-authors, versus they may earn a lot of revenue, versus they may have a hugely wide user base that they can share their instruments with, or maybe a narrow one. They're all very different in what we can see and expect from that. So we try and take a very kind of fair view and measure them on lots of different attributes, and then sort of judge what is the most appropriate targets or aims for that technology versus that technology, is where we're sort of working at the minute.
You may not want to answer this. What's the most difficult technology of the core facility to? Which one doesn't have the same income compared to the others? Doesn't have the same of co-authorship, so the collaborative side, and yet it's still fundamentally vital. But for whatever reason, which one's the most challenging?
Yeah, I probably shouldn't answer that, should I? That's a good question. They've all got their own,
strengths
and challenges, let's say, or opportunities. I don't think there's one that stands out as the hardest one or the most difficult one. I mean, looking back, electron microscopy is phenomenally expensive to run, and that's one factor. Of course, that keeping up with equipment in that space is very difficult. I think if we take a different, maybe a different extreme, is that we have a histopathology facility, which is called the Advanced Molecular Pathology Laboratory, and they can do much kind of higher throughput or much larger amount of samples, and therefore can, their metrics are very different. Same even within microscopy, right? We have some systems that require a lot of dedication to sit down and use and really understand things like live cell imaging, or more extreme, the super resolution techniques. And then we also have a slide scanner, which sits there and just runs automatically the entire time. And if we drill down to the kind of if you look to the financial metrics, the productivity of that versus that is very different. Just because the nature of the instruments are very different.
No, I know. Well, I'm not going to say which one. It's not the most challenging. Not the most challenging to manage. Just their metrics are always, no matter what metric you use, their metrics will be lower than whoever's top. They're never top of any of those metric piles. And that's not the lab, it's just the science.
And it's just the technology, right?
Yeah, and the user base. The user base in some places will be very different. And so actually, labs can change. We still have to have it. It's still an underpinning vital asset. So it doesn't mean the lab's not successful. Just by any metric, it's never going to be the best.
Yeah, that's why we developed this sort of collective of metrics rather than one, right? Because if you look at one, then it's flawed to do so because they may be producing very different expectations and different, things across different metrics very differently. And it's a sort of, balanced scorecard type approach. We're trying to build it now into dashboards so that the platforms, and it's really to help the platforms can put this together and help justify their existence, their funding, their support that they get to the powers that be, and really show the value and the impact and the underlying sort of value they're bringing to the organization through their work, which is, we're preaching to the converted a little bit here, but is of course extremely high and extremely valuable.
Mm-hmm. Okay, switch it for a minute because I know we're getting quite deep into our geek. No, yes. We can carry this on at another point. Some quick fire questions. You're an early bird or night owl?
Early bird.
I suppose it's got to be if you're going out exercising early as well.
Yeah.
PC or Mac?
Oh, Mac, my choice. But I'm on a PC now.
So, McDonald's or Burger King?
Oh, probably McDonald's, I think.
Okay, coffee or tea?
Tea, Yorkshire tea.
We said that earlier. I thought, well, that's a redundant question.
So if my family members come and visit me, they're not allowed in the house unless they've brought a box of Yorkshire tea with them.
And beer or wine?
Beer.
Chocolate or cheese?
Ooh, cheese.
Oh, cool. You're nice and decisive. This is good. Is there any food you don't like?
Not much. I think I suppose if you spoke to my mum, she'd probably say when I was younger I was quite fussy, but now I'm game for trying everything. And I think living here in this really kind of melting pot of all these different cultures and this part of the world, right, is some incredible food options to explore. So I'll try it. I won't say I love it all, but I'll give it a go.
Okay, it's an interesting question from my visit to Singapore. How often do you eat at home? How often do you eat out?
Probably two phases of life here. Pre-kids was eating out a lot more than eating at home. I think post-kids we eat at home a lot more. My wife Charlie loves to cook, so she by choice would probably eat at home and prepare something delicious, as is always the case. Make sure I say that.
So I just the culture in Singapore is far more
eat out. Yeah, and actually, it's a good point. The socializing here is often done over food, whereas back in the UK, it was perhaps more kind of go for a pint or go for a drink. Here, a lot of the socializing is done over food, perhaps less of a kind of drinking culture in that sense, but also the food is a real kind of center point culturally here. It's some of Singapore is very proud of and does very well for sure. And you have these food courts or hawker centers, which are kind of communally shared tables, but with all those food stalls around the outside, so you can kind of pick and choose your favorites from that, which is good ways to socialize.
And what's your favorite food? Do you have a top food?
That's a good one. I suppose so. There's a place here where you can go for satay, chicken satay, etc. And it's a I think it used to be a Glaswegian market that was shipped over to Singapore way back and rebuilt here, called Lau Pa Sat. And every night they close off one street next to Lau Pa Sat and they put out all these barbecues and they start cooking the satay. And it's a great atmosphere. You're surrounded by all the skyscrapers of the CBD in Singapore, and but then there's this kind of lo-fi barbecue going on with great satay. So that's always a great place to bring visitors, and family love it. I think family favorite is, if we said what do we want to go for, I think dumplings would be pretty high up on the agenda at home. For me, I love a good curry as well. That's always a delicious thing to do.
Do you cook yourself?
Badly and rarely, I think is probably fair. I think my wife is much better at that, so I don't do it very often.
Does she work over there?
She does, yeah. She's a teacher now. When we first moved here, she was in medical communications and publications, but she switched a few years back to become a teacher. She retrained, did the PGCEI, and is now a teacher in the international school, one of the international schools where the kids also attend, and is loving it. She's a scientist by background. That's when we met.
Yep.
Back in Edinburgh, and teaches biology and science to secondary school students.
So the opportunities over there have been really quite good, haven't they?
Yeah, it's been a good run, and I think this, like I say, the ability to kind of retrain. I suppose I did that to some degree with studying on top of my biological background. I went and did an MBA as well by distance learning, and I think that has helped also breed some opportunities or helped change direction slightly.
So I'll come back to the quickfire. So I was going to ask about the MBA side, and how much time did that require outside of the day job?
Well, a lot. So I did it. I did it at Warwick University in the UK by distance learning, and this was pre-COVID. So it was done by distance because I sort of selected it based on the reputation of the business school. Being online, it meant I could be and move around and do it if the need arose. And it was a three-year course on top of work full time, and it was a lot of commitments, a lot of late nights, both to attend live lectures and so on at Warwick, but also to cover the material. It was a lot of work, and ridiculously, Otto, our youngest, was born in amongst all that, so there was some very late nights for that reason as well, which was probably a bit of a challenge.
Why did you feel you needed it?
So as we've described, I'd done running a facility. I'd been running a facility for a long time and kind of grown it to a certain point from my own sort of seniority perspective, as well as how it was organized and ran. And I wanted. And I think probably a lot of facility managers would agree. It's a bit like running a small business in a sense that you're in charge of it as a domain, and you've got incomes, outgoings, cost balance, get funding, etc. And some of that I'd sort of highlighted that I was interested in that side of it, as well as I wanted to have perhaps a formal education and experience in how to do it properly, or how to do elements of kind of business administration properly, and to open opportunities to where I could take it next. So chose to do it, and it was great, really sort of enjoyable. I learned a lot, but it was hard work, both for being out of my comfort zone, going back to studying many years after doing it previously, and a completely new topic, right? Being a biologist and scientist entirely by background, it was suddenly doing marketing and doing accountancy, which strangely I enjoyed quite a lot, and all these other disciplines, strategic thinking. But a lot of it has been very useful in running a facility, but also taking the job to where I've gone now, which, as we discussed, is much more kind of administrative and managerial. And
did you pay for that personally?
Yep. It's expensive. I did get some support or a scholarship from Warwick, because they actively encouraged applicants from sort of varied and different disciplines or different backgrounds. So that helped a bit. But yeah, it was funded personally. But because of this requirement to have these diverse backgrounds, or the desire to have these diverse backgrounds, the cohort of students was a fascinating bunch of people, and I think you all had to have 5 years plus experience of managing something or some people, and that brought a real wealth of experience to the conversations. A lot of teamwork and group work as well.
I presume you had to get permission from A-Star to take this on.
I remember at the time, yes, I sought permission from my supervising kind of network, and they were very supportive in doing it in sort of a personal development angle. I don't think, and I hope it didn't impinge too much on my day job. I think I still kept that going well, but and then I used annual leave and so on to go and visit the course. The course required you every semester to go back and do a week in person, which was obviously very valuable. I think I used personal leave for all of those trips. And
more expense.
Also true. I always tagged on a visit home to make sure I sort of said hello to everybody.
So again, though,
I lived down to Coventry. That was part of the problem.
It's kind of in the middle of both of them, isn't it? In-laws and your parents. But again, it's good that A-Star being supportive. And obviously gaining that rewards, because now you're in a different position where those skills and expertise are even more useful.
Yeah, A-Star have been fantastic as a supporting entity and giving me opportunities, I think, shaped by some of the people that I've worked closely with, as well as the organizationally open to me pursuing these things or fulfilling quite different positions to my background. And as I say, we've been here such a long time. It's clearly something's going well, right?
Do you ever see yourself coming back to the UK?
Yeah, that's a question we get asked now and again. We've always said yes at some point, and we've clearly never pulled the trigger on that one. And we don't have a plan, right? We always think about it and talk about it, but we haven't got anything set in stone. I guess with the kids growing up and families getting older and so on, at some point it will become perhaps more real pressure. But for the time being, we're very happy here.
Enjoying it while you're there. Back to the quick
absolutely.
TV or book?
Oh, TV. I'm quite bad at picking up a book and reading it, although I should be better.
Okay. Do you have a secret TV vice, trash TV that you watch?
No, not so much on TV. I get drawn into videos online that are a complete waste of time, but made me laugh quite a lot. So that's probably my vice rather than TV per se. We try and stay tuned into kind of British culture a little bit in the TV we watch, I guess. So watch Have I Got News for You when we can get it, and these sort of things.
And children's TV, I presume, some children's TV.
Yeah. Although the internet and streaming services have changed how that's watched significantly as well. We have much better access to things than when we were first here. Right?
Yeah.
Still watch the odd Man City game and football game as well.
I was going to ask you, what's your football team? So Man City.
Man City, yeah. Which 30 plus years ago, when I chose to support them, it was a decision based on the fact my brother was a United fan, so I thought I'll go the other way. But it was a terrible choice for a while, but it's come good finally.
I don't know if I ever told you I'm a Man U fan.
Well, there you go. There's many Man U fans around though.
Don't forget, I started in the early 80s, late 70s, early 80s, and there wasn't much success around then either.
No, well, so I had to tolerate my brother for a long, long time under Ferguson's years, like late 80s, 90s, of when they were phenomenal, of course. But it's the tables have turned, I'm glad to say now.
Could be turning back again. I like to live in hope. Always an optimist. What's your favourite film?
War. I love the Indiana Jones films, so I could watch them infinitely, I think. So probably one of them. I don't know which one.
The latest one. You must have seen the latest one.
Yeah, the newer ones aren't as good, are they? The old ones, like Temple of Doom, etc., is classics.
Okay. Favorite Christmas film?
I don't. That's a good question.
Do you have one you sit down with the family and watch each year?
Yeah, so I don't know if we've got a solid favourite. I always remember Santa Claus the movie with was it Dudley Moore and stuff like that. That was a classic growing up. Now there's a lot that we watch now. Not sure if we have a family favorite, actually. I think we sort of churn through them all.
Let's see as they get older whether there becomes one that steady down into Star Trek or Star Wars.
Star Wars. Yeah, I know it was never a Trekkie. It never got my attention, strangely. And I know people on this podcast have said otherwise, but yeah, it never captured my attention.
It's pretty 50-50.
Oh, is it? Okay, fair enough.
Yeah. Star Wars, Star Trek is pretty close. And what's your favorite color?
I always say green, and when I think of it, I'm thinking of British racing green. When I say it as well.
See, so why didn't you go fluorescein?
Yeah, could have said that. It's a bit bright though, isn't it?
British racing green. I haven't heard of a fluorescent dye called British racing green.
Quite like yellow as well, but that's probably cycling related these days. Don't get to wear it ever.
Do you have any regrets at work?
Ooh. No, I don't think so. I think in preparation for this, talking to you today, I thought about that and thought about, what I've enjoyed and not enjoyed. I think I've enjoyed it all. I really, really have loved my job at many different times and continue to do so. And have hence continued it and pursued my interests. And yeah, I don't think I have regrets on it. I think it's still good.
And it has so you sent me a picture and you sent me a few things, and one was a montage of lots and lots of people in the community, not just global bioimaging, but all around. And is that a TEDx talk as well?
Yeah. One of the schools here hosted that. It was a good, exciting opportunity. So, this slide sums up, I suppose, the teams that I've worked with, or the outreach and educational and training events we've done as a team. And lots of diverse ones. Right above your head is we did an art exhibition using sort of scientific images that we've taken on the microscopes, an art gallery here in Singapore, giving talks and lectures at different events, whether it's been at sort of the science center or in kind of more informal settings, or at these, like science cafes and so on. And then some of the people like Sarah over your right shoulder, that's hosting a school visit that came and looked around the facility. We do quite a lot of this as much as we can, kind of outreach initiatives, as well as the sort of formal courses like top left and so on. So yeah, it's a kind of drive that we do that, but it's always very enjoyable to interact with people about what we do, or spread the word.
Actually, I should say thank you as well, because the likes of Sarah being obviously to York on different courses as well. So that's quite flattering at that point.
Yeah, we've sent people to the light microscopy summer schools just to, I think when Sarah was new and she joined us, really to get that kind of grounding in education, which starts it for many of us, right? I think that attending a course, for me, it was years ago, Nick arranged Peter Evernett from the RMS to come to Edinburgh and teach one of his microscopy courses, which I was lucky to attend, I think as an undergrad or maybe a summer internship or something. And that, I think these courses or going to get an education are phenomenal for that.
I think they're also good.
Inspiring.
Yeah, not just for learning the skills, but learning how to teach as well.
Yep.
Well, actually,
and get the passion for the subject, right? Like everyone that teaches on these courses they love it, right? And they enthuse about it, and they pass that on to the students for sure.
And obviously, you've named quite a few of the people in here. So you also sent a picture of, I just thought I'd put it on just quickly, lots of the people who you work with over time, the teams that you're working with.
I've been really lucky to have some fantastic colleagues that are captured in many of these pictures that have been fun to work with. Say again?
I love the 3D glasses.
We attended a seminar and someone gave out 3D glasses to everyone in the audience to watch their 3D movies, which I thought was a good delivery method. But yeah, some superb colleagues there that have been part of my team formally or have been associated or linked to us. And it makes it enjoyable, makes it fun, and they've been phenomenal people at doing their job, supporting the microscopy within A-Star and beyond.
So we are nearly up to the hour. I've got a couple more questions I really want to ask you. When has been the most challenging time in your career?
That's a good question. Probably goes back, I can't think of a challenging time in terms of like that, the workload or well, maybe the workload, but anything specific. I think the combination of doing the MBA while doing a full-time job whilst having a baby was probably the most challenging in terms of physical and exhaustion levels. But as I said, I really enjoy what I do and have done, and I've been very, very lucky in that regard. But so I haven't had any real kind of, that was a really tough period of time. I've sort of generally had quite a good time doing it.
If you could do any job for a day, not in the science lab, what job would you fancy trying? You look at all these different jobs out there in the world. Is there anyone you think, I really wish I could just sample it for a day or a week just to get to know what it's like?
I reckon being a cameraman for the latest David Attenborough documentary, whatever that may be, would be superb. Or probably one of the underwater ones by choice, like Blue Planet or something. So I when we were first here, we did a lot of diving, and there's some phenomenal places to scuba dive around this part of the world. But yeah, taking it to that level would be incredible. I think that'd be a fun few days out.
That's a different one to what others have had. So that's quite a cool job to sample. Yeah. What's your favourite conference?
Oh, good one. I think ELMI is a fantastic conference, the European Light Microscopy Initiative, both for the science, for the access to the instrumentation, for the core facilities satellite day. That there's a lot to be said for it, and obviously the community that attends these meetings. I used to go quite religiously, like annually, to make a trip over to Europe to attend, and it was great. Less so now, but we discussed that a little bit. So that to me, that's always been a great meeting, and I would recommend all kind of like microscopists do so, even if their places are limited, as we talked about earlier. The other one is the GBI, the Global Bioimaging ones, the EOE. Yeah. Again, similarly for the network is incredible, and also I've been involved with it not since the start, but for long enough to see it really grow from strength to strength as these more countries have been added, whole new regions of the world have kind of come into that community. And I've been lucky to lead one of the work groups on career paths in that, and that again, coordinating and working with this great bunch of people has been superb.
And that's nearly got a publication, has it not?
Hopefully, fingers crossed. It should be soon. So we published what we call the white paper, kind of international recommendation, which is already available, which is a very long form of the paper. But yeah, we've got it under review at the minute with the Journal of Microscopy, so we keep our fingers crossed, as you did.
Yep. First comments back are promising.
It's looking good.
We'll see. By the time this goes live, who knows? It could actually be in press, which would be kind of cool. And indeed. One final question: in science, is there anything you would change in the way that science works, or within science?
I've never thought about that. What would I change? I think the best bits, or the most enjoyable bits of science to see and do or be involved with is when collaborations are really kind of productive, right? That you can see that two or more people have come around the table and both got ideas and are stimulating and bouncing off each other. So as a core facility, you can sometimes facilitate that and introduce people and kind of make these things happen. That is always a real pleasure and a joy to watch. So if there was more of that, not that there is a lack of collaboration in science, but if there was more of that, that would be great to see. And I suppose the interdisciplinary side of it, as I've been involved in at times in my own work, is really good as well. When you can really put two people who almost speak a different language scientifically together, and yet they can be, really, really thrive off one another. I think that's it's not really a change, is it? Sorry, Pete, but it's something to encourage.
No, it is, because I guess what you're saying is more collaboration, less competition. Competition is healthy, but as scientists, we're competing against time, and so we don't need to compete against each other. There's always that urgency to get results out. So the competition is internal, and to be competing, repeating, and collaborating.
Yeah, I guess I wasn't coming from that angle necessarily about the expense of competition, but just I think it's where the magic happens a little bit sometimes when different disciplines come together, or different even research groups, right? There can be really some valuable things in there. And I think with microscopy, you see that whether it's the physicists or the bioinformaticians or the data scientists, that you get this marrying of capabilities is very good.
Right, so on that note, Graham, I should say thank you, and thank you for everyone who's watched or listened. Please subscribe. Do you know what? I don't think anyone has name dropped so many previous guests. I kept telling here. We had Stefan, we had Timo, we had Kota, we had Jon Eriksson, we had Jan Ellenberg, Antje Kepler, and I probably missed one or two others that you've name dropped. But there's loads to catch up on the series. Graham, you've also been so easy to talk to. You kind of talked your way through it. It's just been really great, and I hope for those listening, you understand a different career track and some of the nuances and the potential to develop a career out of that. And I think I got lucky at you've obviously got very lucky at A-Star, and a very supportive community within that as well. So Graham, thank you very much.
Thank you very much. It's been an absolute pleasure. Thanks.
Thank you for listening to The Microscopists, a bite-sized bio podcast sponsored by Zeiss Microscopy. To view all audio and video recordings from this series, please visit bitesizebio.com/the-microscopists.